Carol. Thanks, Kelvin. So my job today is just to introduce the topic. You will notice in this talk that it is going to be just some fundamentals of erection physiology and how Botox may have improvements in corporal blood flow and innervation. I'll make the point now that the putative mechanism by which this occurs is not known, but I'll provide you with some hypotheses and some data to support it. I think the way that I'd like to frame this question is to discuss preclinical modeling and to talk about erection physiology and how preclinical models help us determine and delineate how new therapeutics can potentially treat ED. And then we'll move into Botox and how this influences the corporal microenvironment and neurotransmission. This is the erection physiology as we know it. This is not a truly comprehensive, but it makes the point that you've got a regulatory balance between vasoconstriction and vasodilation. We're all aware that the NO cyclic GMP PDE5 axis is the main neurotransmitter responsible for vasodilation. And we know that the sympathetic tone, norepinephrine, cholinergic innervation, and I would say Rho kinase and Rho A, Rho kinase signaling is responsible for contraction. So with that in mind, let's transition into how this may work. I'm going to use the model of cavernous nerve injury as a basis to explain how Botox may work. We're very aware that the innervation to the penis, the parasympathetic, sympathetic, and non-adrenergic, non-cholinergic innervation comes from the spinal cord through the pelvic ganglia and then to the neurovascular bundle in humans. This is actually a rat with the major pelvic ganglion, and if we use models where we crush the nerve to mimic that which occurs in radical prostatectomy, this also occurs with peripheral denervation as it relates to diabetes. We see that there's profound changes in the end organ. This is actually an example from a radical prostatectomy. This is actually human data where a group from Italy actually took biopsies of the penis 3 months after patients underwent a radical prostatectomy, and you can see here that there was both an increase in collagen deposition as well as a decrease in smooth muscle. So you're seeing a balance between a reduction in smooth muscle and then increase in fibrosis, which leads to some of the corporal veno-occlusive dysfunction that we see in prostatectomy. So what happens with innervation influences the end organ penis. If you look at models of nerve injury, and once again you see this in diabetes, you see that there is a reduction in neurogenic or neuronally innervation to the penis. And if you try to stimulate the nerve after nerve injury, this is just an example of what happens after 48 hours. You have a reduction in intracavernosal pressure, which is how we measure penile blood flow into the penis after nerve stimulation. And you can see that 14 days after nerve injury, we have a reduction, but then over time, upwards of 60 days, you see that function improves to nerve stimulation, suggesting that there is a regeneration or an improvement in neuronal function. So this is the basis for how we study erection physiology. So the question is Botox really a potential therapeutic target for erectile dysfunction? So how is it administered? It's administered into the penis locally. You inject it into the penis, and then you can measure indices of penile blood flow by duplex Doppler ultrasound. You can also obviously questionnaires and see how this affects patients with different forms of erectile dysfunction, which we're going to hear in our debate today. So what are the consequences of Botox injection in, for example, in another field? Okay, so the expected effects of Botox into a muscle is that muscle is actually going to be weakened due to neuromuscular dysfunction or block neuromuscular transmission. So it is blocking neurotransmission to the skeletal muscle in this case, or to the corporal smooth muscle in our case. What are the unexpected consequences of Botox, for example, into a skeletal muscle? Well, it blocks neurotransmission in both the cholinergic synapses, but also in other synapses, in particular the sympathetic, so norepinephrine, substance P, and the like. There is selective weakening and depending on muscle activity. So you actually have a weaken the muscle. And if we all understand, and probably a lot of people in this room use Botox for bladder dysfunction, right? This is sort of the basis of that. And then you see that when you inject, there is clear signaling to the spinal cord, which goes into the central nervous system via retrograde transport. And this has significant plasticity in the central nervous system, how that relates to a peripheral denervation. And there is some thought that actually Botox may actually be neuroregenerative. So with that in mind, how is Botox thought to work in the penis? Well, I'm summarizing a handful of articles. Let me make it very clear. There is, I would say that there is no clear understanding of how it works, but these are the hypotheses of how it works, and I'll provide you with some data. It is thought that it acts by inhibiting acetylcholine, norepinephrine release at the presynaptic cholinergic junction. So this would result in a reduction in smooth muscle tone. It increases cyclic AMP, not cyclic GMP, which is thought to be what we know is the main mediator of vasodilation, with that of increases in PKG. And we, at least in preclinical models, Botox actually has never been shown to have effects on non-adrenergic, non-cholinergic nerves. This is the neuronal nos, or the nitronergic nerves, right? So there is actually data from, and I will acknowledge, it comes from the stomach, from the fundus of the stomach, which shows Botox has zero effect on this. However, there is some thought that it may in the penis. Botox reduces vascular resistance and allows for an increase in penile blood flow via the mechanisms I just showed you, and it is thought to have neuroregenerative effects. First, I'm going to start with a model of spontaneously hypertensive rats that had developed erectile dysfunction. This is from Francois Giuliano from a couple of years ago, which looked at Botox in combination with PDE5. I started with this paper because it is looking at the effects of Botox on nerve stimulation and blood flow into the penis. What it shows is that Botox, when given 1 week prior to nerve stimulation in spontaneously hypertensive rats that have erectile dysfunction by the parameters we study, has an effect, both a direct effect with monotherapy and a synergistic effect when combined with a PDE5 inhibitor, in this case sildenafil. This is an abstract that was presented at our meeting, actually a couple of years ago, by Jeff Campbell, which looked at neuronal nitric oxide synthase and alpha smooth muscle immunostaining in the penis in a cavernous nerve injury model after Botox injection. So look at that middle figure there for nNOS. When you have a sham animal, so this is an animal that did not undergo nerve injury, you see an increase in neuronal nitric oxide synthase, suggesting that this may have an effect on nitronergic expression. And we see that, but you do not see that in the nerve injury model, suggesting that you need an intact neuronal innervation for that to occur. But he did see an effect on smooth muscle actin in both those that were denervated as well as those that were intact, and he saw an improvement in erectile physiology. So in conclusion, I think we should strive for the best translational science. I think we have a lot more to learn from our preclinical modeling as it relates to Botox. And then I think the proposed mechanism of Botox in the penis is under investigation without clear understanding of the physiological benefits in the penis. And I look forward to hearing about the clinical data in the debate next. Thank you.